So let's look at the pancreas. Now, this is a look at the islets of Langerhans, right? Or the islets of Langerhans. I think in Unit 2 biology we do look at histological comparisons, so I inputted some. So the endocrine pancreas, we have an exocrine function, but we're looking at only the endocrine function. The exocrine function would have been to produce certain types of enzymes, right? But we're only looking towards the endocrine function at this time, right? So the endocrine pancreas consists of clusters of cells called pancreatic islets, right? Or the islets of Langerhans, right? So I'll be speaking to them as the islets of Langerhans, right? So these islets contain different types of hormone secreting cells, primarily the alpha and the beta cells, right? So let's have a look at the pancreas now, of the interlobular duct, right? That all of these interlobular duct is the way in which the hormones and different things pass through the pancreas, right, into the bloodstream. Okay. All right. So we know where the pancreas is and everything, where it's attached to that C-cell biology. Right? We're moving towards what Kate is asking for now, right? So we have a look at the islet of Langerhans, and we have some alpha cells, some beta cells, and some delta cells. But we're looking at alpha and beta in Kate biology, right? So the alpha cells are located in the islets of Langerhans, right? They produce glucagon, right? The function of glucagon. What's the function of glucagon? Go ahead.
It breaks down glycogen into glucose.
All right. So we have the production of glucose via gluconeogenesis, right? So it promotes gluconeogenesis. Lovely, right? But the beta cells, which are also in the islets of Langerhans, and they produce insulin. What is the role of insulin?
Insulin converts glucose to glycogen.
Right. So not only does it facilitate the uptake of glucose back into the cells for polymerization, right? But it inhibits gluconeogenesis, right? So alpha cells would have increased blood glucose levels, while beta cells would have helped to decrease blood glucose levels. Let's look at that, right? The regulation of the blood glucose concentration, right? So stimulus rise in blood glucose levels after eating. What is the response? Right? Our beta cells will now produce insulin into the bloodstream. Insulin promotes uptake of glucose by the cells, particularly in muscles and adipose tissue and also the liver. Right? Glucose is either used for immediate energy use or stored via polymerization to produce glycogen in the liver, and muscles are converted to fat for long-term storage. Right? That's what's happening with high blood glucose levels. For low blood glucose levels now, we have decreased amount of blood glucose, right, in the blood. What is the response is now? Our alpha cells will now take it upon themselves to produce glucagon, right? Right? Stimulating the breakdown of glucose via glycogenolysis, right? And the synthesis of glucose from non-carbohydrate sources, gluconeogenesis, right? So listen to this now, right? Glycogenolysis is usually what occurs because we usually have cells storing glycogen, right? But remember here what insulin did? Insulin can convert sugars into fat. So gluconeogenesis would occur now when we're synthesizing glucose from these non-carbohydrate sources. Gluconeogenesis, right? And this will raise the blood glucose level. All right. So we're looking at this. All right. So this is looking at all three of the types of cells, but unfortunately, alpha cells and beta cells are the only ones we're looking at, right? Looking at insulin and glucagon. All right. So this is the feedback loop. All right. Let's have a look at it. Go through it And obviously, if there are any questions, rather, please ask. Any questions? All right. If there are no questions, that is it for our understanding of the pancreas with its blood glucose concentration regulation. All right.